Professor Jared Rathel here. Thanks for tuning in to this second installment in the evolution of populations. So, short lecture here. What I'd like to do is hone in on those three factors that drive the vast majority of evolutionary change, ultimately sculpting exquisite adaptations like the false eye spots on the giant silk moth that you see on your screen. So we're still thinking herein about microevolution. Of course, microevolution are small changes in allele frequencies from one generation to the next. So we're thinking about what disturbing factors push us out of the Hardy-Weinberg equilibrium and actually cause our allele frequencies to change from one generation to the next. The three major drivers that cause the majority of evolutionary change on planet Earth are 1. Natural selection, 2. Genetic drift, and 3. Gene flow. And we'll talk about some special cases of both genetic drift and gene flow at the end of this lecture. And of course, we know all about natural selection. We remember our avian predators, those hawks and owls, taking out light-colored rock pocket mice on dark lava flows in New Mexico's Valley of Fire. You remember that activity from earlier in this unit? Here on this slide, we actually see the opposite selection. So this is, again, an avian predator. This time it's a great gray owl, the largest owl species in North America. And this time it's snatching a dark mouse, easily seen on white snow. So let's take a moment and think about how selection can manifest itself. And we'll go ahead and we'll stick with mouse color phenotypes. So at the top of your slide, here is your ancestral population of mice. We've got 5 phenotypes ranging from light coat colors to tan to dark. And we see this nice bell-shaped frequency distribution where the more common morphs, the medium tan morphs, are the more frequent, as indicated by this red curve. Okay, so recall the Valley of Fire in that rock pocket mouse activity. We had these dark lava substrates, right, created 1,000 years ago. And we had selection against lighter phenotypes. So what happened is the distribution of the original mouse population, there's the original mouse population, it's illustrated here with the solid red line, it's shifting now in one direction. So it's shifted to the right towards those darker phenotypes. And then here you can see the new distributions are the dashed line. So this is an example of what we call directional selection. Directional selection can go either way. In the Arctic, like on the slide you saw before, where the substrate is covered by snow most of the year, you can envision the directional selection shifting the other way as white mouse phenotypes are selected for. And so again, all that is driven by predation and differential rates of survival. Okay, now let's look at the middle panel down here. Okay, disruptive selection. I want you to imagine a beach, maybe in the Hawaiian Islands. So you've got light colored sand all over the beach, and then you've got these emergent patches of dark volcanic rock. So you could see how an environment like this, a substrate like this, might create disruptive selection, where the once rare phenotypes, the light-colored mice and the dark-colored mice, now become proportionately more common, where the dark colored mice are selected for on the dark rock outcroppings, the lighter colored mice on the sand, and the result is you've got this new bimodal distribution. You've got two dominant phenotypes, right, on either side of the distribution. So this is creating disruptive selection. And then the final example of selection, stabilizing selection, this occurs when one phenotype provides such a powerful selective advantage. It's just hands down better than the other phenotypes. And what happens is the alleles that code for the other phenotypes are dramatically reduced, as you see on the bottom right there, the stabilizing selection. Just one phenotype is exhibited. So taken to the extreme, stabilizing selection can actually fix the phenotype, where 100% of the individuals, 100% of the mice express, in this example, the medium brown coat color phenotype. And the other alleles that code for light and dark, those might be lost from the gene pool. So natural selection, obviously a major driver of evolutionary change. Genetic drift refers to the radical change that we can see in allele frequencies from one generation to the next when the population size gets really small. And then chance or randomness, sometimes referred to as stochasticity, has a much, much greater impact on the population. Why would chance play a larger role when the population gets small? Consider a coin flip. If I flip a coin 10 times, it would not be that improbable if I got 7 heads and 3 tails, even though we all know that a coin has a 50-50 probability and we would predict 5 heads and 5 tails. If you have just a sample size of 10 flips, a small sample size, it's not that unlikely to deviate from our predicted 50-50 outcome just by chance alone. Now, imagine I take that same coin and I flip that coin 10,000 times. If I got 7,000 heads and 3,000 tails, you would claim, and rightly so, that that is a biased coin. When sample sizes or population sizes get large, we approximate expected outcomes. But all bets are off when population sizes get small. Genetic drift can cause radical change in allele frequencies from one generation to the next when the population size gets small, and it can induce rapid evolutionary change. A great example of the detrimental effects of small population size is the Florida panther. The Florida panther is a subspecies of mountain lion whose population was so small, only about 20, maybe 30 individuals in the mid-1990s, that inbreeding resulted in all of these genetic abnormalities, defects like undescended testicles in the male and heart defects. In 1995, 8 mountain lions from Texas, which is a different subspecies, were moved from Texas into the South Florida area and took up residence with their Florida cousins. How could just 8 of these panthers, these translocated panthers, save a whole population? Well, those 8 Texas panthers brought with them new alleles. And this genetic rescue counteracted the negative effects of small population size, reduced genetic variation, and inbreeding. And as the population numerically rebounded, there's approximately 300 Florida panthers now, the influence of genetic drift waned. So the Florida Panther is a success story where conservation biologists exploited the benefits of gene flow. So genes flowing across space from one region to the other, it's a huge driver of evolutionary change. Think about a handful of anole lizards out to sea, floating on a log, colonizing a new isolated island. Those founding individuals, or that one pregnant female that wrapped it over on the log, the alleles that the founding population happens to carry, those alleles are likely to be very different proportionally than the allele frequencies in the original ancestral stock. This is what we call the founder effect. When a small number of individuals, sometimes just one pregnant female, splinters off from the ancestral population and founds an entirely new population, rapid evolutionary change is likely to follow. And you're going to be thinking about Caribbean anole evolution on your virtual lab activity coming up later in this unit. An example of the founder effect in humans occurs in the idyllic farmlands of eastern Pennsylvania, home to an Amish culture that originated from a small group of German immigrants, probably like 200 people, that settled in eastern Pennsylvania in the 1720s, 1730s. So at least one of those original 200 founders carried an otherwise rare genetic allele that coded for a disorder, causing dwarfism and polydactyly. Polydactyly is a sixth finger or toe, like you see on the right-hand side of the screen there. Compound this, Amish marry within their own community, right? So a good Amish boy is going to marry a good Amish girl, and you don't have a lot of new genetic variation that's entering this population. Today, there are some 50,000 Amish that are living in eastern Pennsylvania. All of those folks descended from about 200 founders. It's a closed population. And today, the rates of this syndrome that causes dwarfism and polydactyly are much, much higher than the Pennsylvania population at large. Why? The founder effect. When a small part of the original population moves to a new locale, from Germany to the New World, or when the population is reduced to a really small size because of environmental change, the genes of the founders of the new society are disproportionately frequent in the resulting population. The founder effect. And one final example of the dramatic impacts of small population size on allele frequencies is the bottleneck effect. A population bottleneck is a sharp reduction in the size of the population due to some catastrophic event, like an earthquake or a flood, fires, droughts, famines, disease outbreaks that spread across the population. So most of the original population, like you see in the bottle here on the left, right, with all of this genetic diversity. The genetic diversity is represented by different colored marbles, where the marbles are different alleles. And the original population's got lots of different alleles. So most of the original population doesn't survive. Just a handful of individuals make it through the bottleneck. And that handful of individuals carry alleles, just happen to carry the alleles that they have. In the future, the population may rebound numerically, right? It may grow large again in population size, but the genetic diversity, the diversity of different alleles is now greatly reduced. And genetic diversity is going to remain low, increasing only when gene flow from another population occurs, as did with the Texas Panthers moving in, being translocated to South Florida, or very, very slowly over time, over millennia, as random mutations occur. This new population, it's now less robust in its ability to adapt to and survive in a changing environment. It has less genetic raw materials to cope with a new virus or a rapidly changing climate. Cheetahs are the textbook example of the bottleneck effect. So at the end of the last ice age, at the end of the Pleistocene, about 10,000 years ago, cheetahs almost go extinct. So the entire global population of cheetahs probably gets down to less than 100 cats. Today, in 2020, the cheetah has numerically rebounded. There's about 7,000 cheetahs left on planet Earth, but the remaining cheetahs have very low genetic variability. They are highly inbred. So you may know that genetic differences among humans means that when we have an organ transplant, like a kidney transplant, the recipient has to take these really powerful immune suppressing drugs to keep our bodies from attacking that new kidney because our immune system thinks it's a foreign invader. Even if the kidney came from a sibling or a parent, with cheetahs, they are so genetically similar that you can take a skin graft from a cheetah in Botswana, here at the southern end of their range, and you can graft it to a cheetah in Iran, and you're not going to see an immune response because these cheetahs are so genetically similar to one another because of this bottleneck that they went through 10,000 years ago. Okay, in conclusion, we talked about the three primary factors that cause the vast majority of evolutionary change on planet Earth. One, natural selection. We talked about directional, disruptive, and stabilizing selection. We talked about genetic drift, small population sizes, as well as gene flow, and some special examples of both small population size and gene flow, the bottleneck and founder effects. So right on. I hope you learned something. I appreciate you tuning in, and I'll see you next time. Thanks.